Students, now we will see about another application of biotechnology that is used for production of transgenic animals. So far we have seen the application of biotechnology in agricultural field, in medical field, and then in molecular diagnosis, that is in medical field, and now we will be seeing that is production of transgenic animals. What are these transgenic animals? Animals whose genome is manipulated or altered to possess or express foreign genes are known as transgenic animals. Getting? Like how genetically modified plants are present. Okay? Like this only, these are genetically modified animals. So other name for genetically modified plants is transgenic plants, right? Similarly, there are transgenic animals, means whose genome is altered to express foreign genes. There the plant genome is altered to express the desired traits in the plants, to get the desired flower color or to get the maximum yield. We have seen different advantages of GM plants, that is tolerance to abiotic stresses, okay, enhanced nutritional value, maximum utilization of minerals. These are some of the advantages of the GM plants. These transgenic animals are nothing but they are genetically modified animals known as transgenic animals. Even transgenic animals have different advantages at present, we will be seeing. So these are nothing but they are the animals whose genome is altered to express foreign genes, or it is altered to possess the foreign genes, okay, which is going to produce the desired product or the desired function in these animals is known as transgenic animals, right? Such as rabbits, rats, pigs, sheep, cow, fish. There are different types of animals which are genetically modified, known as transgenic animals. So the rats, transgenic mice is present, rabbits is present, sheep, cows. These are some of the transgenic animals. And transgenic animals are produced with different techniques. So either by inserting extra functional gene in the embryo, or gene is introduced in such a way that is going to knock out the specific gene, that is specific target gene will be knocked out, and therefore the gene which is externally inserted, or the foreign gene known as transgene, will be adapted by these organisms, right? So the different methods to introduce the foreign gene and to produce the transgenic animals, there are three different methods. One is DNA micro injection. So what is DNA micro injection? These are the techniques we have studied in biotechnology principles and processes, that is these are the techniques to make the cells competent to take up the foreign DNA or exogenous DNA. So this technique is used to create transgenic animals, okay, that is we are going to introduce the recombinant DNA, or you can either introduce the foreign DNA directly into the nucleus of fertilized egg to produce the transgenic animal, right? In case of plant cells, to make genetically modified plant cell, usually the technique used is gene gun method or biolistic method. It is an important neat bit. In order to make the transgenic animals, the DNA micro injection is the widely used technique to produce transgenic animals. So in DNA micro injection, it is a direct injection of the recombinant DNA or the foreign DNA, or you want to introduce a functional gene, okay, using micro pipettes or micro tips or micro needles, or the capillary tubes are used to introduce directly the gene of interest into the nucleus of the fertilized egg. So as the when you are introducing it in this stage, so when the this fertilized egg undergoes repeated cell division and develops into a offspring that is going to possess the all the cells will possess the foreign DNA or the transgene that is going to express the desired function. right? This is one of the method to create the transgenic animals. Another method to create the transgenic animals is retrovirus mediated gene transfer. That is retrovirus mediated gene transfer. Why? Because retroviruses are used as cloning vectors to clone genes in the animal cells. To clone genes in the animal cells, retroviruses are used. Why? Because retroviruses contain RNA as the genetic material, and they have the capability to infect the animal cells by which process? By a process known as reverse transcription. So using that, retroviruses are going to transform normal cells into the cancerous cells. So based on that ideology, once again the retroviruses are used to transfer the foreign gene or the functional gene into these animals, okay, known as retrovirus mediated gene transfer, to create transgenic animals. Means that animals that is going to possess new traits, or they are going to produce new products, or they are going to produce the products at very high yield and large production area, right? Another example or another way of creating the transgenic animals is embryonic stem cell mediated gene transfer. Another way to create the transgenic animals is embryonic stem cell mediated gene transfer. We have seen one is DNA microinjection used for animal cells, whereas in case of plant cells, gene gun or ballistic method is used. And retrovirus mediated gene transfer in animal cells, whereas if you have seen the BT cotton and parasite nematode resistant plant, they are genetically engineered using agrobacterium mediated gene transfer. You should know these differences, student. And here we are using embryonic stem cell mediated gene transfer. Once again, the cells in the embryonic stage are genetically engineered, or they are inserted with the foreign gene, okay, or gene knockout system, wherein you are going to insert the functional gene at the embryonic stages by removing the non-functional gene, that is resulting in gene knockout system. Means a specific target gene is removed and the gene of interest is replaced in that particular location, that is known as gene knockout system. And if that process is done at the early embryonic stages, and that too in the stem cells, because when as the fetus develops, the stem cells will multiply, and that is going to produce the different cells and tissues and organs. So all these cells will possess this foreign gene, or the external functional gene will be present in all the cells of the newly born offspring, right? For that purpose, this foreign gene must be inserted in the embryonic stem cells, okay, using a gene knockout method. That is going to help in the whole organism, that time will be completely containing the foreign gene in its genomic DNA. So these are the three types or three ways to create the transgenic animals. Now let us see what are the advantages of these transgenic animals. Why we need these transgenic animals? What does these transgenic animals do? Right? Why we need transgenic animals? Why we modify? In order to get some benefit from these animals, we are going to modify to produce certain vaccines or to produce certain antibodies. Yes or no? The transgenic mice are used to study certain diseases, or they are used in monkeys are used to test certain vaccines. So there are different applications of these transgenic animals. If you want to study any disease, because human life is precious, we cannot use the human life for testing purpose, right students? Nobody will be interested, apart from certain volunteer people, no one will be interested to give their life for testing these vaccines or medicines and all. Who knows how the vaccine is going to react? Yes or no? For that particular purpose, we require transgenic animals or transgenic models. So the transgenic animals can be used to study normal functioning and mechanism of the living system. So let us see some of the advantages of the transgenic animals. One of the advantage of the transgenic animals is to study the normal physiology and development. One of the advantages to study the normal physiology and development. Now you might be knowing what is normal physiology and what is pathophysiology, right? That is studying the normal functioning and mechanism of a living system, like how the genes involved in regulation of these functions, or how these are formed, and how the development of organs, cells and certain biological compounds is going to take place, and how they interact with one another. Right student, see, in our body many biochemical reactions are going to take place, and each biochemical reaction is mediated by an enzyme, right? And every enzyme once again is secreted by certain type of cells. Okay, so the whole body is running with a in a network like area, right? Means one is interacting with another one, and everything is going in a balanced way. If there is any abnormality taking place in these biochemical functions, okay, because one metabolic product pathway, whatever is producing a product, will be precursor for another metabolic pathway. Okay, so everything is interconnected in the body. So we should know how these biological compounds are present and how they are interacting with one another for the cell growth and development, how they inter connecting with the cell growth and development. So that is nothing but the normal physiology and development. If one gene is coding for a particular product, that is a protein is formed, if it is formed properly in the required amounts, then the whole process will be smooth and the end product will be formed. This end product is going to be precursor for another biochemical pathway, right student? So therefore studying the normal functioning, it is normal physiology and development, is very very important. So in order to study this normal physiology and development, okay, the transgenic animals are decided or they are designed, that is going to express and regulate the foreign genes, and we are going to study how they are going to interfere in the normal body functions and development. Okay, if you are inserting one foreign gene directly in the human body, and you are going to observe the variations taking place in the human body. Okay, if the variations are disadvantageous, then what will happen? It will lead to the death of the human body. That is the reason for any particular disease, if you want to study a disease, okay, how a disease is caused, how it is affecting the disease genes, why the disease is caused, in what conditions this gene is behaving as abnormal, or in what conditions the product is not formed properly. Okay, all these studies can be done in the transgenic animals. So that is studying the normal physiology and development. To study the normal biological pathway of the human body, we require transgenic animals. We are going to insert certain foreign genes in those animals, and we are allowing them to express in these animals through their gene expression system, and then we are going to find out what are the effects of it, what is the studies, whatever is required are observed in these transgenic animals. So one such example for this normal physiology and development is studying the growth factors. Okay, where here transgenic mice is used, okay, as a transgenic animal to study the growth factors on the growth of an organism. Okay, by introducing certain study specific genes that is going to lead to the formation of these factors. What are the factors that are used for studying is, example, insulin like growth factors are used for studying the growth factors, how these growth factors are helping in the cell growth and development. Okay, how these growth factors are affected by other proteins and other compounds, and how the expression of this growth factor is going to takes place. Yes or no? Now in any some cases, if the physiology is becoming pathophysiology, means something is getting affected there. Okay, that is studied in the transgenic animals. So one of the example we are going to consider is insulin like growth factors. What are these insulin like growth factors? They are peptide hormones. Insulin like growth factors are nothing but they are peptide hormones, and they resemble in similar in structure, okay, and functioning to that of the normal insulin. Here we know one insulin hormone is produced by beta cells of pancreas, right? That is going to have the action in the decrease in the blood glucose level, whereas its major function of the insulin growth factors is in the stimulating the growth factors. That is going to stimulate in the growth factors, which is going to responsible for the cell growth, differentiation of the cells, fetal growth, as well as the development of the fetus. This is once again it is stimulated by these insulin like growth factors, which are similar to the insulin in the minor functioning, but the major function of these factors is not decreasing the blood glucose level, but they are going to stimulate the growth hormones, okay, or growth factors, and that is such as platelet derived growth factor, nerve growth factor. These are some of the growth factors that is going to help in the cell growth and development. Okay, so these growth factors are made to express in the transgenic mice is used for these studies. These are genes are altered and they are expressed, either they are less expressed in the transgenic mice and their effects are studied, okay, or they are overexpressed and their effects are studied. Why? If that expression is taking place in the human body, how it is going to make a difference compared to the normal? What is the abnormal condition will be if it is over expressing or if it is less expressing? What would be the difference are studied, right? So these insulin like growth factors, their major function is to stimulate the growth factors, or it stimulates the growth hormones of the pituitary gland, and these growth hormones are going to stimulate the liver to produce IGF-1. There are two types, one is IGF-1 and IGF-2. Okay, these insulin like growth factors will stimulate the growth hormones of pituitary gland, and these growth hormones will stimulate the liver to produce IGF-1, which is very important, okay, whereas IGF-2 is less important in stimulating linear growth, but IGF-1 is very important, okay, as it is going to stimulate or increase in cell size. When IGF-1 is produced by the liver, okay, it helps in the cell size increase as well as cell number increase. You can observe the amount of these IGF levels in the humans, okay, in the childhood. These IGF-1 factors, okay, they are present at very high levels because that is a development age, okay. So these IGF-1s are required for the increase in the cell size and cell number for the growth of the individual. So therefore in the childhood these IGF levels are very high, okay. As they are reaching towards the puberty, they are at very peak, okay, and once they are moving towards the adult stage, these IGF-1 factors are going to be reduced or they are going to decline, because there in that age the growth is not going to be that much rapid and it is going to remain stationary. So in the childhood age it will be increasing, and either at the puberty age it is going to be at peak, and as they are reaching towards the adult age these IGF-1 factors are reducing. So in the transgenic mice these IGF-1 factors are introduced in such a way that they are going to overexpress the IGF-1 factor, and they studied its effect on the postnatal brain growth because it is helpful in the postnatal development after the birth of the baby, okay. They are very much important in the growing stage of the brain as well, as well as it is developed in the fetal growth also. It is very much necessary. So they have introduced in the transgenic mice and they have overexpressed this IGF-1 factor, and they studied its effect on the postnatal brain growth. So they have studied, they have seen the result as there was an increase in the brain size in the transgenic mice that is expressing high levels of the IGF-1, that is expressing high levels of the IGF-1, and they also studied the effect of another factor, that is IGF-1 binding protein. Okay, IGF-1 binding protein that is acting as a inhibitor to the expression of IGF-1 growth factor. Right, IGF-1 binding protein is going to act as inhibitor for IGF-1 growth factor. So therefore, they have observed that these binding proteins are inhibiting the growth release of the IGF-1 factor, and that is going to retard the brain development, leading to brain growth retardation. So they have studied now, okay, in the undernutrition cases what was happening, there was a high or the production of this IGF-1 binding protein that is going to decrease the IGF-1 levels. So they have, once again the research was made and they tested in the transgenic mice. Okay, they have overexpressed the IGF-1 factor, and they have created a gene that is going to suppress the IGF-1 binding protein, so that it is not going to affect the gene coding for IGF-1 factor, and the results were seen as there was the improvement in the brain growth retardation during the early postnatal life. Okay, if any individual is suffering from this brain growth retardations at the early postnatal stages, there are the chances to develop the brain, okay, by overexpressing this IGF-1 factor. Okay, in some cases, in some cells, if this IGF-1 is not produced properly, then we can externally insert a gene using genetic engineering technique that is going to express this insulin like growth factor 1, that is going to help in the development of the child at its postnatal life, early stages of the postnatal life. Right, students? So this is one of the example to study the normal physiology. Means now we know which gene is coding for the growth factor, and why this growth factor sometimes is not expressing properly. It is because of the another inhibitory protein, that is IGF binding protein. So this link is studied through the transgenic animals, especially transgenic mice, and the report is generated that if this IGF binding protein levels are decreased and overexpression of this IGF-1 growth factors takes place, then we could develop the brain retardation problems, or we could prevent these brain retardation problems and improve the brain development. So this is one of the example of studying the normal physiology and development using transgenic mice. Another example of using transgenic animals is study of diseases. It is study of diseases. So how we are going to how it is this transgenic animals will help to study the diseases, right? Transgenic animals, especially mice, have been extensively used as a model for various human diseases, because these models are resembling almost 99% to that of the similarity to that of the genetic composition of the humans. Okay, and they are used to study pathological mechanism behind the disease and the genes responsible for development of disease. Based on these studies, new treatments for disease are discovered. Getting students, you can study what is the pathophysiological mechanism that is taking place. Okay, what is the gene that is present in these diseases that is helping in the causage of the symptoms and all can be studied using the transgenic animals, especially transgenic mice. So some of the diseases in the humans such as cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's disease as well as cancer are studied in the transgenic animals, and they have tried to find out what is the reason for the causage of this disease, what are the genes that are responsible for causing this disease, and what is the mechanism. Okay, if the gene is expressed in such a way, what is the biochemical changes, or the what are the changes taking place in the physiology are studied. Okay, and then what could be the treatment for this particular disease is also studied using the transgenic animals. So in cystic fibrosis, how the cystic fibrosis is caused? If there is a mutation in the particular gene known as cystic fibrosis transmembrane conductance regulator gene, that is CFTR. Students, in cystic fibrosis, there is a gene known as CFTR. Okay, that is cystic fibrosis transmembrane conductance regulator gene. This gene, if it is mutated, then it will cause to the disease condition known as cystic fibrosis. The mutation gene is going to produce the dysfunctional CFTR protein. Functional CFTR protein will be formed when there is a normal gene coding for cystic fibrosis is present, then CFTR protein will be formed. If at all there is a mutation in the gene of CFTR, that is cystic fibrosis transmembrane conductance regulator gene, then whatever the protein CFTR protein formed will be dysfunctional. And what are the functions of this CFTR protein? It is going to act as chloride ion channel, as well as it is going to help in the fluid transport system in the epithelial cells. Okay, that is going to help in the sweating as well as the removal of chloride, and it is going to maintain the water moisture system in the body. Okay, if there is a mutation in this CFTR gene, then it will code for a dysfunctional protein that leads to the accumulation of mucus. You can see the patients who are suffering from the cystic fibrosis will have a sticky mucus. Okay, that causes accumulation of mucus in the lungs and other organs, leading to complications such as respiratory disorder as well as the diabetes. Okay, so it is a genetic disorder because of the mutation in the gene coding for CFTR protein, that is going to result in this cystic fibrosis or this genetic disorder. So the studies are done in the transgenic mice in regard with this cystic fibrosis disease. Apart from that, in the rheumatoid arthritis, so it is an autoimmune disorder, okay, causing painful swelling and eventually resulting in the bone erosion as well as in the joint deformity. Okay, and we have Alzheimer's disease. It is loss of memory and inability to carry out day-to-day activities. This is also caused because of the overexpression of a amyloid protein precursor. Okay, it is caused due to the overexpression of amyloid protein precursor. This is going to form plaques or neurofibrillary tangles in the nerve cells. So the expression of this gene that is coding for amyloid protein precursor is studied in the transgenic mice. Okay, when this gene is overexpressed in the mice, it developed plaques and tangles in the transgenic mice, as that it happened in the human beings. So associated factors affecting the accumulation of this amyloid is also studied in the transgenic mice, and the actual reason for these symptoms is found out and treatments are designed accordingly. So the treatment also, they are going to make a drug will be developed, and it is in first the disease condition is introduced in the transgenic mice. Okay, the genes coding for this amyloid precursor, there is a substance known as amyloid protein precursor. So accumulation of this, excessive accumulation of this results into plaques in the nerve cells. So when there is something disturbance in these nerve cells, the nerve cells will not transmit the signals properly, because of which there is a loss of memory and inability to carry day-to-day activities. Okay, so this particular gene is inserted in the transgenic mice, and it is allowed to overexpress to form the plaques. First, the disease condition is created in the transgenic animals, and then different drugs are developed and they are injected in these transgenic animals, and they have studied how it is going to affect, how long it is going to treat these plaques. Okay, so this is also studied in the transgenic mice, as well as the cancer conditions are also studied in the transgenic mice. Okay, and then we have another two applications or advantages, that is in the production of biological products, vaccine safety testing, toxicity testing. These are also are the advantages of transgenic animals.